What is the cell danger response? One of our research aims is to test that hypothesis that all chronic diseases are organ-selective manifestation of the cell danger response. The cell danger response is ultimately controlled by the conversation between mitochondria, the nucleus, and neighboring cells. This happens because mitochondria are located at the hub of the wheel of cellular metabolism. All the air we breathe and all the nutrients we eat and drink are ultimately delivered to mitochondria to help us move, think, work, and play. Mitochondria continuously monitor the chemical environment of the cell and instantly respond to danger by changing their activities from healthy function to cellular defense. When cells go to war, they do what nations do when they go to war. When the CDR is activated, cells harden their borders, don't trust their neighbors, and restrict the exchange of resources with their neighbors. New discoveries in our lab have traced one of the root signals for maintaining the CDR to purinergic signaling. Cell danger response biology. The new science that connects environmental health with mitochondria and the rising tide of chronic illness by Dr. Robert Naviaux. The cell danger response is a universal response to environmental threat or injury. Once triggered, healing cannot be completed until the choreographed stages of the CDR are returned to an updated state of readiness. Although the CDR is a cellular response, it has the power to change human thought and behavior, child development, physical fitness and resilience, fertility and susceptibility of entire populations to disease. Mitochondria regulate the CDR by monitoring and responding to the physical, chemical, and microbial conditions within and around the cell. In this way, mitochondria connect cellular health to environmental health. Several studies now report the presence of dozens to hundreds of man-made chemicals and pollutants in placenta, umbilical cord, blood, and newborn blood spots. New methods in metabolomics and exposomics allow scientists to measure thousands of chemicals in blood, air, water, soil, and the food chain. Systematic measurements of environmental chemicals can now be correlated with annual and regional patterns of childhood illness. These data can be used to prepare a prioritized list of molecules for congressional action, ranked according to their impact on human health. So you have the CDR response, and it can lead to all of this. Most recent publications. This paper shows that organisms can become hypersensitive to their own extracellular ATP. This makes them react more quickly and forcefully to stresses in the environment that cause ATP to be released from stressed cells. It also shows that many abnormalities that are commonly measured in children with ASD, like carnitine and fatty acid oxidation abnormalities, changes in microbiome metabolism, chemokine and corticosterone response, the fever response, and even plasma vitamin levels are each controlled by ATP signaling. Another key finding was that females are more sensitive to the metabolic effects of extracellular ATP. Males are more sensitive to the behavioral effects. These findings are relevant to ASD, ME/CFS, PTSD, post-treatment Lyme disease syndrome, major depressive disorder (MDD), and the risk of suicide in teens and patients with ME/CFS.